Good afternoon. Mage tikde. Good afternoon. Okay. It is my absolute honor to introduce our distinguished speaker for this second session, Dr. Deepak Lokwani sir. Sir is presently working as an associate professor at Department of Pharmaceutical Chemistry, Rajeshri Shahu College of Pharmacy, Buldana, Maharashtra. Sir has more than 15 years of research and academic experience. His expertise lies in organic, medicinal and computational chemistry. He is skilled in molecular modeling, chemoinformatics and proficient in designing virtual screening of new chemical entities using variety of CAD techniques. Dr. Lokwani sir earned his PhD from Pharmacy Department of Chemical Technology, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad in 2016. During his doctoral research, he was awarded Senior Research Fellow from ICMR and worked as a project fellow on UGC major research. Sir has excellent research record where he has published more than 60 international papers in high impact factor journal with a cumulative impact factor of 198 with a H index of 22 and over 1200 Google citation. He has authored 6 books in pharmaceutical organic chemistry, computer-aided drug design, and contributed 3 book chapters to edited book published in international publisher. Sir played an active role in scientific publishing as a guest editor for a special issue on a computational synthetic approach in drug discovery in a journal of molecules. He has received national prize for short film in a CSIR open source drug discovery and Vigyan Prasad short video competition on the need of new drug for tuberculosis. For his outstanding publication, he received a national award from V Life Sciences Pune for his contribution in computer-aided drug design and molecular modeling. Now I request sir to please take charge of the dais and start his session. Please sir. Am I audible? Last benches? Am I audible? Yes or no? The mind is full? Stomach is full. Yes or no? I'm not audible. Last one. Please, please. Yeah, that's great. Great. So, first of all, thank you to the managing principal of Srinath College of Pharmacy for inviting me for a session, right, on the AI and drug discovery. So we are going into some part that AI is, okay, what the different methodologies involved in the AI, what are different tools which we be going with the AI we can work out, okay. Before going with that, I want to tell you that the one thing is that particularly the AI. Now you all of you know, but you have done about the AI session in particularly in the first one with the Lahoti sir, right now? Yes? So sir have told you the number of things with respect to AI, what exactly the AI are, right now? No, but the AI means is basically with simplicity your mobile. All of you have a mobile? Yes? Yes? Now in your mobile, there is a lock. The screen lock is there, that have a fingerprints are there, the face are there. So now how it's working? Is it working automatically? No. But you are giving something first we call the input. And that input is nothing but we call the preparation or data. So you are providing the data, right? How you are going to provide to the data to your mobile? You want to see that. Of this one. Now think that if it's my mobile, I will give my mobile to my face. Okay?
Now after giving, the mobile will, that AI system will say that this is the face of a Deepak. Oh, you have to recognize, this is the Deepak, right? After the recognizing, he remember the face for the number of duration of time till we will change it. Now, but if somebody else coming to just open up my mobile, that mobile should have to provide, or we should have to provide my face or my face only to open it, right? Otherwise, you cannot open that particular mobiles, right? Similar way, the AI system is working in terms of a drug discovery, in terms of other way. Please, it can handle by my side? Yes, please. So, in terms of a drug discovery, particularly the AI is work on in a different categories. In terms of, we can call the different phases of drug discovery starting with the target identifications. So, target means what the target is basically a disease. The disease can be working based on, we call protein, based on the enzymes, based on the receptors. Now, take the example of your COVID. The COVID is basically started with the spike proteins, the M-pros, and the number of enzymes are there, which will be excreted and enter inside our body, and they create a particular infections are there. So what we have to do, we have to target, we have to find out the targets. So how the AI is helping in the target identifications. So the overall, in these sessions, we are going to learn about the different targets, what the targets are, how we are going to identify this target with the help of AI, okay, and what the different tools are available in AI that will be help us to identify this one. So this can be helpful not only to the pharmaceutical chemistry students or the teachers, but can be helpful for pharmacologists, can be helpful to the pharmaceutics, helpful to the other categories of, we call the pharmacies. Now, it's not only the disease selection or target selection, but it can be helpful for the drug discovery or hit identification or the lead identification. Now, we want to design some molecules. Number of molecules are there, we want to design it. So how we are designed? So previously it is a CAD. We call it as a computer-aided drug design. But now this CAD is totally, we call overcome by the advancement of AI. And AI have a lot of subsets. So the overall we are going to learn about what these subsets are. But in terms of this one, why I keep these slides for drug discovery, because before going with the AI, you should know what is the drug discovery. So drug discovery starts with the disease, drug discovery, and with the drug in the market, right? So start with what the disease are, what are the different targets in the disease are, and how we are going to identify the new drug for that particular disease, and now how this drug is modified or optimized in a different way, and then how it is going into the market. There are a number of steps. So in each stage, we are using the AI. We are using the AI, right? Now the AI can be anything, particularly with respect to these tools. So the AI, before going with the AI, but what the computers have started the first journey, basically they come in 1964, where you almost know about the QSR. Anyone knows? Yes? That we call the quantitative structure activity relationship. That almost it done in 1964 by the Hansch, and then after in 1960s we have a CDs computers are came out. Then in 1980s we have a chemoinformatics are in a market out. Then we have in 1990s the number of softwares are around, and one of the software is basically the Schrodinger. You know all of you, right? Now, in terms of Schrodinger, the chemistry students of chemistry faculties very well know about the Schrodinger, what the Schrodinger is doing, what the different tools are available with the Schrodinger. But the Schrodinger is almost a commercial software. Now, I'm not going with your commercial software. I'm going with your open source softwares, open source tools, which we can help us, not particularly for the chemistry, the pharmacology, even with the pharmaceutical students, or the teachers to do the research, to identify the drugs, produce the drugs, to formulate the drugs, and all this we are working. So it's almost in 1990 we have a Schrodinger's, we have a OpenAI systems are there. In 2000s we have a CDKs there. In 2004 the ligand databases are started, and almost in 2020s we have artificial intelligence that we call as AI. So AI is not started immediately. Take almost 50 to 20, 60 years to come out with the help of our computers and all, and help of our data we needed. Understood? Now, what I am saying, again I am on the same part, that mobile, we are giving our face, we are giving our fingers, Whenever you're providing the data, you should have an output. If you're not providing the data, you should not have any types of output. जब भी हम लोग एक पर्टिकुलर डेटा देंगे तभी हमें आउटपुट मिलेगा, otherwise we are not getting any types of data. So AI is basically working on the data only. You should have to provide a data. So whenever you have experimental data, you can get an output. If you don't have experimental data, you should not have output. So that's why the AI has taken a time to become out, and that's why in the 2020s we have AI, and that is only because at the time of a COVID and all, everybody is rushing for the new drug for the COVID, but we not find out any drug because the COVID. In particular, that year, we not have a particular what exact the targets inside the COVID, what exact is the different enzymes or receptors of, or particularly the that the mechanisms of that COVID is working. So at that time, the computer systems has evolved. They have created a lot of data for us. So we will see one by one exactly what this. So what exact AI? The AI is basically a technology allow the computer to perform the task, and that should be equivalent to the human. The task which we call the human intelligence are using. They are not exactly the doing by the computer, but what we are providing to the computer, that will be done by that computer itself. So in terms of that, this task can be the problem solving, the task can be the learning, the task can be the decision making. So always have to remember, whenever you are going with the computer, you have to first learn out the computer. You have to do this. You have to do this, that multiple times you have to do this, and then computer will do that. Not simply you have to say that about the AI. We have a data to learn, we have a data to reasons, we have to self-correct. With the data, we have to collect the data from the different resources, and AI can use the logic and decision tree to solve these particular problems. Now, but particularly the AI. The AI have two subsets. The first subset is called as a machine learning, and second subset is called as a deep learning. Artificial intelligence, the machine learning, and the deep learning. So these are the three different parts. And in terms of a drug discovery, in terms of a drug designing, in terms of a drug formulation, we are mostly using the machine learning and the deep learning as compared to AI. Now, all of you are using the Chat GPT. Yes, everybody is using. Even the faculties, the teachers, everyone in India, everyone in the world is using the Chat GPT. So what exactly the Chat GPT is directly coming something? No, it is behind something. Models are there, and what these models are? These models are the text models. Something the models are given by the scientists, input given by the scientists to that particular computer, and based on the computer they have developed a model, and from that model it is going to predict a new text that we call as a text related model or text related AI. But what the machine learnings are? The machine learning is simply the picture is given. The input is car. Input is car. Now the computer is putting the car is there and providing some data behind we call creating a model behind it, and output will be the car. Okay. So whenever you're giving the car to the computer, always output is a car. But if you're giving the bike and model is based on the car, that model will ignore it because the input is because that input is basically a bike, not a car. That's the difference between machine learning and that particularly AI. AI is basically after development of model with respect to the human intelligence. The machine learning is basically have to learn the machine first. You have to provide the input and learn out the machine what exactly you are going to do, and that based on that you are getting the output. And similarly, we have a deep learning. Deep learning is advancement of a machine learning that we call the number of layers is going in the background. Now, I just simply want to say that although to develop a model, right, for the pharmacy is not an easy task. It's basically you need a person from mathematician, you need a person from a computer science background, you need a person from a biological background if you want to work on a drug discovery. So these three persons along with the pharmacy can set and they can create a model. But alone of pharmacy you cannot develop as a model, because for the model developing you should need the knowledge of a computer, you need a knowledge of a Python language, you need a knowledge of a commands and the codings. Without that, you cannot create your models. But as a pharmacist, people, we can use the already developed model for our sake of R&D. So just what I am going to say you, or I am going to cover here, basically first what the artificial intelligence, what the machine learning is, what the deep learning is, what are the different methodologies involved in a machine learning, and how we are going to create our task in a machine learning, and the different tools are available there. What the tools are there with where we can apply our knowledge, or where we can provide the input so that we are getting the output. Now take the example, I am working on one drug molecule, but I don't know what is a drug that is suitable for any particular patient or not, because it has a lot of toxicity. Right? So maybe there possibility. I am working on one molecule, I am going to synthesize that molecule, but that molecule might be toxic. So how am I going to predict it? I have to work on the animal and I want to check its toxicity. It will take a lot of cost. I need the animal, I need the time, I need the we call the resources to perform the activity. But if you have a model developed by the AI which can predict the toxicity, so that you can predict directly by using AI. That model can be based on the machine learning. That model can be based on the deep learning. So there the AI have two subsets. The first one we call as a machine learning, and second one we call as a deep learning. The deep learning is basically working on the neural networks. Machine learning is based on the providing the input data. You recognize the input data based on the identification of that data. Identify that is my face, that is thing, that the car, that is bike, that is machine learning. But the deep learning is basically working because of your brain, the neuronal system. Similarly, the machine learning. Inside our brain, there are a lot of neurons. They are working automatically. Right? Whether we are controlling it? No. Our unconscious mind is controlling it. Similarly, inside that model, deep learning model, there are a lot of the methods are there, a lot of algorithms are there, which are creating such a model which is unable to be called handled by our mind itself, and that's why we need the computers. And the computer, and all of you know that computer will just learn about the 0 and 1, right? There is only recognizing the 0 and 1, and for the sake of this one, they recognize the 0 and 1, you have to provide the data always in a 0 and 1. You not have to provide this is the car. It not recognize the car, it recognize it is 1, and recognize the bike as a, okay. So this is what have to provide the input. So we are going again, what exact will be there. So machine learning is basically have a 3 steps. The first step is to provide the input data. To provide the input data. But from where we are getting the input data? Anybody knows? From where we are providing the input data? You want to create your model. Now take the example, you want to create your chat GPT. From where we are getting the data down? To provide the almost a text of a historical data. Now consider, I want to work on creating a chat GPT, or want to create a my chat GPT on a historical knowledge only. So I have to first provide the all historical knowledge to the computer. This is the history. This is the history of we call the Sambhajinagar, and that we have to provide to computer. Now, whenever you ask to the computer again regarding that history of Sambhajinagar, you will give answers. So you have to provide, first one is called as a data. So in terms of drug discovery, you want to create a model with respect to toxicity, respect to ADME properties. So you have to first provide the experimental data with respect to ADME. So whenever you are providing the data with respect to ADME property, you are always getting an output in terms of the ADME property. But if you are providing the data with respect to synthesis, you are not getting a data for the ADME property. So the input will be respect to the output. So what you are providing, and then with respect to that what you are getting. So in terms of this one, you have a first one we call the data collections, and the second one is data preparation. Now data preparation is basically have a actual what we call the machine learning will work. There are different data. They are working on the different models. One of the model is called as a expansion models. Now in terms of expansion model, if you providing a data called A, it automatically expand to the data B. It automatically expand to data C, and then it automatically expand to data D. So there are a lot of different models with respect to the machine learning. These are again, it's a part of a computer science. It's a part of what we call the chemoinformatics persons. It's a part of what we call a person handling the Python language. But as a pharmacist, we are going to apply it in something else. So we are not going to bother about what exactly they are doing. We are bother about what data we are going to provide. And lastly, there should be have to develop the model, and that should be have something with respect to we are getting. Now see that in the first one, The model is providing the form of a picture. What is called the cat and what we call the dog. The input is given. Output, what we are getting the output is a dog. It is recognizing the dog, and whenever you providing the dog in there. Now, in the same time, in terms of we call here you are getting a laboratory data. So that laboratory data we are providing from our laboratories. We are providing from the different sources. These sources are available. Lot of sources are available where you are getting a laboratory data. Now this data can be in the form of structures. This data can be form of biological activities. This data can be in the form of ADME properties. This data can be in the form of if you want to perform the electrosynthesis, that we call the synthesis, that molecule which you want to synthesize. So this data can be anything that we call as the laboratory data. Now ultimately, the ML model will be developed. Now this one the ML model is developed, you have to apply it for the prediction. Again, in terms of justification with respect to that is called the machine learning, provide the input data, prepare a model, okay, provide the input data, the statistical model will be developed, and then the prediction of something, the part you needed, that we call as a application of this machine learning are. So, in terms of that, the first part is basically the data. From where you are getting the data? For getting a data, you have a lot of resources are there. So one of the resources is basically called as a zinc. Zinc is almost contain the millions of molecules which are available from the procurement. They are available from the different sources. These structures are basically in the form of the different formats are available along with the biological activity. The form of we call the novel molecules. They are available there. So you can download these all the structures if you want to create your data. If you want to create your we call it as a model. Okay, so that is one of the zinc is there, and one of the another data database is called the Campbell. Campbell database is basically a curated, manually curated database with a bioactive drug like small molecules. It contains the binding, the functional, and additive properties. That again, with similar to the zinc, this database is more better with respect to zinc. Here, you are getting a biologically reported databases where the biological activity is already reported by the yourself. Now, consider you are publishing of papers. So when the papers are published, they are going into the database of some publishers. What the Campbell will do, Campbell will collect your database, collect your paper from there, collect your structures from there, and store into their repository. That we call the Campbell. So this is the we call input data. Now we have an input, we can download from the Campbell, and we can prepare our model from the then. Similarly, we have one more database that we call as a Hopkins. It's similar thing. Campbell is also one of the free we call houses of millions of chemical structures with their biological activities. It provides the 3D conformers as well as the physiochemical properties. It is integrated with the toxicological as well as the pharmacological data. Apart from that, if you want to work on some drug related material, now take the example. All of you know about the drug repurposing? Yes? Anyone, please? What is drug repurposing? Another use of a same drug or already reported drug already available in a market that we call as a drug repurposing. So you know any drug which is repurposed? Aspirin. It's well known example we have. The aspirin is basically analgesic, but we are using for the blood thinning, and that is mostly we are using particularly in a diabetic. Now similarly, we have repurposed the drug lot of time during the COVID. Have you remember the examples? Have you remember the example? We have used lot of drugs during the COVID. Your chloroquine is there. Yes. Yes. The remdesivir is there. Right. So lot of examples are there. We have used lot of times in a drug repurposing. So similarly, if you want to create the database, want to create an input file for the already US FDA approved drug, then you can download the database directly from the drug bank. So drug bank is available. You can download the all structure directly from here, so that you can create your model. Okay, so you can create the what we are going. We are going to the first step from where we are going to get the data. So you can get the data from the pumpkin. You can get the data from the zinc. You can get the data from the we call Campbell. You can get the data from the drug bank. Similarly, you can get the data from the binding DB. It is also one of our database which is containing the measured binding affinity of a protein ligand molecules. Similarly, one more database there, which is a commercial database. It is called as a CellChem. Now you can, but CellChem is also contain the target specific database. If I want to work on the neurological disorders, so I will get a database related to the neurological disorder. If I want to work on a diabetes, I want to work on a glucokinase activators only. I am getting a database from the we call the CellChem. If I want to work on only COVID-19, or I want to work on only particularly any enzyme, so I get a database from the CellChem, and we can download it and we can arrange the model with respect to that data. So this is the first stage in terms of from where we are getting the data. In second stage, what we are doing? We are arranging this structure in the form of a computer language. What is a computer language? 0 and 1. What is 1? 0 and 1. Computer cannot recognize your structure. Computer cannot recognize your carbon. Computer cannot recognize your oxygen. It cannot recognize anything with respect to or without that we call the 0 and 1. So to notify this what we call the structure in a 0 and 1, there are the different formats, and one of the format is called as a smile. Is it visible, everyone? This is called as a smile format. So the exact will be there. I just put up the molecule with that we call the paracetamol. Structure of a paracetamol, which is basically contain a benzene ring connected with the OH, and then it is connected with the NH and COCH3. So just the carbon number 1 and carbon number 2. They are available in we call capital. This capital carbon is nothing but belong to the aliphatic carbon. So whenever you're writing a smile format is basically a text format of your structure. The text format of your structure. I'm not talking about its chemical name. I'm not talking about its molecular formula or molecular weight. I'm talking about how to write a structure in a text form. Text form. So that is basically called as a carbon. The two carbon which are available here is basically aliphatic carbon, which is belong to CH3 and C. CH3 and CO. And then it's a bracket. The bracket is indicating the branching. Brackets are indicating the branching, and there is a then double bond, and there is a O is there, right? And then it is given about we call N. And then it is a small c with 1. Small c is 1. The small c is basically of a aromatic structure. And 1 indicate the where aliphatic chain is connected or from where your carbon atom is started. That's why it is a 1. So capital C indicate about the aliphatic. The branchings are indicated by the brackets. The double bond itself is a double bond. The O can be the O, and then the aromatic should be the small. So now that is C. How many C's are given? 3 C's. 1, 2, and 3 C's are given, and this is 1 C, second C, and this is third C. And again there is a bracket. Because there is a branching started because of OH. That's why these two C's are basically in a branches, and again it is put up the C1. Now C1 you have to start it. C1 you have to end. C1 we are starting because the C1 is and C1 we have started and C1 we are there ending. They are connected to each other. The connectivity they are defined. So that is the completion of a ring. Because the 6 carbons are there. We can put up the C1, C, and again C1. So they are the 6 carbons. They are connected to each other. That's the format we call as a smile. Now once you find out the smile is there, you have to arrange in a form of we call it the zeros and 1. Which form? Zeros and 1. To arrange this form in a 0 and 1s, we have to provide different nodes, and one of the nodes is called as the 1 hot encodings. There are the different ways to arrange it in a 0 and 1. There are a lot of models of our AI. We are not going to cover all model in a single lecture or in a single session. Although there is a course in an AI to complete it. Even one subject is not easy to complete in an AI. So one session is almost not possible there. So we want to go with some models or some way we can arrange this text form in the form of a 0 and 1. So what we are, we form a matrix. Simply a matrix. So in matrix what I have done, this smile we arrange in a y-axis and the x-axis. Okay. So now the C is connected with the C. Yes, it is 1. The C is there, so there are two C's and two capital C's are there. That's why I given the 1 and 1. And no, this is the bracket. So C is the bracket? No, that's why it's a 0. Again, that is plus sign and C is same? No, that's why it's a 0. So the wherever on x-axis, on y-axis, same letters are coming, you put up the 1. When the x-axis and y-axis, the different letters are there, then we call it as a. Now to create such 0 and 1 model for all the structures, manually it takes a lot of times. We can provide to the computer millions of molecules, millions of structures. Computer will do it within hours. Because we are putting the languages or background behind to the. Okay, that's the difference. If I want to create this slide, it takes me the 10 minutes to create a slide, to draw the structure, to convert the structure in a smile format, and to convert a smile format to the one hot encoding. But a computer is an easy task, and this is the second stage in terms of machine learning. First one, to give a database. Second one, to create the language as per the computer needed. Now similarly, I have put up one more example. There is an example of a graph notation. Graph notation. Now simply see that if my fingers are same, no, right? This finger is small one, this is bigger one, again small, this is again a small. This is a representation of a graph. So every graph has some nodes and edges. Now if I draw a square, there is a graph, there is a edges and the nodes. So ants are considered as a edges. In 2 points, there is a bonding is considered as a edge. Sorry, there is the points are considered as a nodes. Points are considered as a nodes. The bonds are considered as a edges. Similarly, you see that this carbon is considered as a node. It is connected to the OH. OH is also considered as a nodes, and this bond is considered as a carbon considered as a node. Second carbon is considered as a node, and this bond is considered as a. So this is called the graph. In terms of graph, we have a nodes and edges. Nodes, edges, connectivity. Okay? Or now, in terms of same, we have a x-axis and y-axis. X-axis and y-axis, but something different from your encoding and the graph. In terms of we call it as a graph notation, C1 is connected with the C1? No, because it's the same. That's why it is a 0. But again, C1 is connected with the C2? Yes, C1 is connected with the 2. That's why I put up is a 1. Is C1 is connected with the C3? No, that's why 0. C1 is connected with the N4? No, that's why 0. So, based on the connectivity, whenever C1 is connected with the C2, we put up the 1. In a chain form, in a ring form, C1 is connected to the C2, we put up 1. C1 is connected to the C6, we put up 1. But when C1 is not connected with the C4, so that's why I put up the 2. So this is we call as a graph notations. So this is the second way we can put up the data to the computer. But we have to provide the data in the form of only 21s. We are going to provide the data other way. And now, once we are providing the data to the computer for the machine learning, what a computer will do? Computer will develop the models. Artificial intelligence will develop the model, and this intelligence will develop the model in the base of the machine learning or the deep learning. Now, I'm going to do the 2 part of only machine learning, because the machine learning have a lot of different way to arrange this data to create the models. Once we are giving the 0 and 1s, it created models. But there are the different way to create a models. That is, we call a we call basically super visualized we call learning, the unsupervised learning, we call the reinforcement learning, the deep learning is there, the neurological learnings are there, the number of learning is there. And one of the learning which we are using in terms of ChatGPT, that we call the natural language processes. So there are a lot of different way particularly AI is creating a model. But in terms of machine learning, I'm going to cover the 2 ways by which we can creating a model. The one is called as a supervised learning. In terms of supervised learning, first you're providing the data in terms of 0 and 1, right? You already provided data. Now, after providing the data in a 0 and 1, you should have to give its label. You have to provide it along with the label. This is 0 and this is 1. Now, the computer will recognize this. So, this is 1. This is carbon, this is oxygen. This is 1 is indicating about the connectivity. Okay, this 1 is indicating this is about the this atom. So similarly, I am putting up one picture here. This is the label data. So there are the three different labels are there. That is, one is called the hexagon, and one is about the triangle, and one is about the for the squares. Now you are providing the data from the came. You download lot of the structures, or you download it from the drug bank. In drug bank, almost it contains 7,000 US FDA approved drugs. Whether they belong to same category? No. Whether they belong to same pharmacophore? No. Whether they belong to the same chemical structures? No. But how the computer will recognize it? Right? But first you have to provide in 0 and 1s, you have to provide in a graph notation, you have to provide in encoding forms, and then you have to provide a label. That this types of encoding is belonging to the analgesic drug. This types of encoding is belonging to the antiviral drug. This types of encoding is belonging to the some other categories of drug. Now the computer will recognize it and memorize it. Whenever you are providing a new drug, what the AI will do? AI will just overcome about their database. उसके पास में जो models रहेगा उसमें वो check करेगा कि किससे match होता है. From where it is going to match? Whether it is matching with the antiviral, whether it is matching with the analgesics, and then predict, oh, this is the drug which is matching the analgesic, so that this drug should be analgesic. Okay, so similar way here, whenever you providing the label data, that label data can be hexagon, right? That can be the triangle, and that can be the square. Whenever you're providing to the AI, this model will prepare a model in terms of recognizing the label. And once when you are providing the taste data, now you're providing the taste data in form of two. One is a square form and one is a triangle form. What the computer will do? Separate. Square is different and triangle is different because you're providing the data, providing the form of a label, and such label is called as a supervised learning. Now second learning, we have a unsupervised learning. Now there's an animation is going on, but the input data is containing the strawberries, the bananas, and the grapes. Right? We are not providing here the labels. We are not going to provide the labels. In last, we are providing the label, hexagon, the squares, and triangle. But here time we are not providing the label. But the unsupervised learning means if the computer will create a classification, computer will create a cluster. What a cluster means? A group of similar peoples. Now in terms of audience, we can create a group of similar peoples. The students are in a second year. The students are in a third year. The students are in a final year. Some are in the MCOM. Some are the faculties. This is called as a classification. What we calling? Classification based on some categories. Right? Similarly, your model, what it will do, it create a classification. The classification can be based on the structures, similar types of structures. Classification can be based on the similar types of connectivities between the structure to structure. Classification can be based on a similar types of we call as a pharmacophoric features. Classification can be based on the similar types of a biological activity. So whenever it create a classification, it create a cluster. These two we call method which we are mostly using in terms of pharmacy to segregate the data or to create a models. One of the model is based on the supervised and one of the model is based on the unsupervised data. This is we call as a developing a model that we call as a machine learning. Now, once this is done, you have a lot of complex system with you, right? And finally, you are providing the data and you have a output. Based on that, already I think Susar have covered number of parts from the drug discovery, what the ML, what the AI will doing in there. But the AI have a lot of we call applications in each where the drug discovery. The first one in all category, actually I'm going to cover the one tool in each category. But with respect to drug discovery design, with respect to the polypharmacology, with respect to we call chemical synthesis, with respect to called drug repurposing, with respect to called as a drug screening. So whenever you're talking about the AI and drug design, the AI can be used to predict the 3D structure of a target proteins. Now all of you know that inside our body is everything is a protein. We are working only because of a proteins lot is doing inside our body. Whether it's a biocatalyst is working, whether the receptor is working in a single we call it induction or in terms of a single transaction is there. Okay, so then number of things are going inside our brain, and that's because of protein. So we should have a protein structure with available. Now the predicting the drug protein interaction, so that you basically call it as a molecular docking. In terms of chemistry, students or people are working, right? Now, it's not only molecular docking is useful in the chemistry, but it's also useful now in the pharmaceuticals, because you want to notify what exactly the interaction between your polymer and your drug molecule, what exactly the interaction between your drug and your solvent, what exactly the interaction between your drug and the lipid, what exactly the interaction between the two proteins. So everywhere, when you want to predict something, the interaction, you have a mind at terms that is called as a molecular docking. So the AI can be work in molecular docking also. It can be used in predicting the drug activity. If you want to predict the activity, you can generate a model, you can predict the activity. Similar like again a chat GPT, if you're providing the text, you're providing the prompts, you're getting a something output. So similarly here, if you're providing a something structure, the AI will give you what exact its biological activity, what exact its solubility, what exact its ADME properties. So what you want, the AI will give you based on that drug design. Similarly, one is called the pharmacology. Everyone, what about the polypharmacology? Anybody? Polypharmacology is again a similar terms with respect to a drug repurposing. Poly means a single drug, multiple activity. That we call as a polypharmacology. So similarly, we have a AI and a chemical synthesis. Now, a lot of times in terms of industries, lot of times in terms of a pharmaceutical chemistry peoples, they are suffered or they are working on how to synthesize a new drug. If they are getting a new drug from the designing point of view, what is the synthesis, because steps it is involved. AI can also use in retrosynthesis. That means you can a final product you have, and from the final product you can break down in a such a way so that you're getting a reactant molecule. Rather than from the reactant to product, what we are going here, from product to reactant side. So that is opposite, that we call the drug repurposing. And lastly, we have a drug screening. The AI can be useful in terms of prediction of toxicity, in terms of a bioactivity, in terms of predicting the physiochemical properties. So this is all about we call the something the AI we have. Now we are going with some tools by which they are available open sources and we can use for our studies either in terms of any particular with respect to the field. But if you want, you can use it in a free, is available. So one of the them is called as the AlphaFold. Can we know about anyone? The AlphaFold. The recent news about the AlphaFold is coming 2024 itself. Anybody please? AlphaFolds. Listened about this term? No? So this is one of the websites which help us to predict the protein, predict the three-dimensional structure of protein. Now, how many structures in terms of proteins do you know? The proteins are basically made up of amino acids, right? But these amino acids are basically called the primary structure of protein. A primary structure is basically a two-dimensional way you can draw on the board, but this actually not inside our body. Inside our body, the protein is available in the form of we call three-dimensional or in a we call as a more than this a four-dimensional will be there also. The quaternary structure we call either in a tertiary structure or in a quaternary structure. So that we call as particularly the primary structure first converted into the secondary one. And the secondary structures are basically of two types. One we call the helix and one we call the beta strand. Whenever I draw the protein in the form of the we call the angles, something like a coil shape that we call as the alpha helix. When I draw these amino acids in a parallel, anti-parallel sides, we call the beta helix. So this beta helix and this particularly we call the beta strands and helix combine to each other, they form the tertiary structure. So this tertiary structure can be predicted based on only giving the sequence. So it's not possible till now. But with respect to this alpha fold, we can predict easily the tertiary structure. You just provide the sequence of a protein, or you just provide the name of protein, it will give the protein structure in a three-dimensional form, which you can use for your molecular docking purpose, you can use for the protein-protein interaction, you can use for your biological work or any other particular effect. And for this sake, particularly the one of the company that we call as a DeepMind and its scientists have recognized with a Nobel Prize in 2024. And now, just now, this DeepMind is overcome by Google. Google has purchased this particular company, and that's overcome with that we call now the Google on AI company, which is called the AlphaFold. And basically, there are the three scientists who are working on this concept. One is called the scientific Davis, the John Jemper, and the David Baker, have recognized with the Nobel Prize in 2024 just for predicting the protein structures. So before that, we can identify the protein structure with the help of AI simply. Actually, the protein structures are basically developed or identified in an experimental way after the X-ray crystallographic structure or after the NMR. But now it can be predicted just by providing the name, just by providing the sequence. You just go on this particular site that we call as AlphaFold, and you provide us the name of a protein which you want, you're getting a protein structure. Just search on the Google, go on the AlphaFold, just provide us the name of any protein, you're getting a protein structure. After getting a protein structure, it gives a protein structure in a three form, in the form of a confidence. If that confidence is very high, the structure can be good. The confidence is not good, then that itself AlphaFold will say it's a bad structure. And then third form is basically we call it an ugly structure, a very, very low confidence is there. Because what they are doing, they are particularly recognize the structure and superimpose that structure with respect to the protein data bank. The protein data bank itself is a repository where these proteins are available, or actual three-dimensional X-ray crystallographic protein structures are available. So from here you are also going to get them. This is about the AlphaFolds. Now similarly, with respect to AlphaFold, there are the number of other methods or other servers are available. The links are given also there. This particular link is coming from the GitHub. The GitHub is basically a repository of number of tools with respect to AI. So you can go on the GitHub, just search about any types of repository to get it. Okay, whether it's respect to again chemoinformatics, whether it's respect to your called network pharmacology, whether it's respect to molecular docking, whether it's respect to we call the molecular dynamics simulations, or whether it's respect to the ADME property. Just put up the name there, getting a lot of AI, and okay, so just we are going fast. So now another one is basically we call it predicting the drug and protein interactions. So with respect to that, you all have to know about the docking. Yes? Anyone? No, what is a molecular docking and all? What is a virtual screening? To perform the virtual screening, you have a perform a steps. From where you're getting a target. Okay? You're getting a target and you're going to dock a molecule there. Going to dock a molecule there, and after the screening you're getting a hit molecules, right? So this is a virtual way by which we can perform this one. So this one can involve a QSR, this can involve a pharmacophore, and based on that we have a lot of tools are available. So I'm going to share the slides, you can see the tools, but one of the tools I'm going to share you with this called the Veena. All of you know about the Autodoc Veena? Yes? Yes? All of you know? Yes? Anybody have handled Autodoc Veena? Any chemistry students? Yeah, that's great. So you handle the Autodoc Veena. But now Veena is basically not working on the AI. It's simply working on the Monte Carlo algorithms. What the algorithms are? The algorithms are basically a mathematical equation, a mathematical way by which we can recognize a molecule. Now you consider this is the software, this is the protein. A protein, and you have a simpler structure. It is going inside a protein and it performs a degree of we call interaction. One we call as a rotation, a translation is there on the basis of we call as a x-axis and y-axis as well as z-axis, and then it recognizes whether the shape of your ligand exactly match the binding pocket of a protein. So this is we call as a Monte Carlo algorithm, which is working on the expansion. But the Veena is basically the advanced part of your Veena. It's a fork of a Veena, and the fork of that we call the Veena, which is working on the deep learning. It not only provides the scoring from the Veena, but it's giving the extra scoring on the basis of a deep learning. So in terms of a molecular docking, if you're working on that one, rather than working on the Autodoc Veena, go to work on the Veena where you're getting two different scores. The one score is basically with respect to your binding affinity. What exact the shape is matching of your ligand with the protein. Okay, what exactly is matching with each other. And another score is basing on the we call the deep learning based on the input data of you are provided to them. Okay, so now this is about the Mina. Similarly, the one more type software or tool is that is called as a deep docking. All these tools are free. You can work out, but you have to be a expertise or at least a proficiency in a command line, because these all softwares are working on the Linux. They are not working on the particularly the Windows way, because the Vina can be work on the Windows, but when you want to work on the AI system, you should have something knowledge about the Linux, a small knowledge. I'm not saying you should be expert in a Linux system, but at least know about what the commands are and how to work on. And one of the software is called as a deep docking. In terms of deep docking, what it will do, it will dock the billions of compounds. Now, if you want to dock a single compound, it will take a 5 minutes. Consider, if you want to dock a single compound, it take a 5 minutes. And if you want to dock a billions of compound, how much time it will take? Maybe 15 days, 1 month, right? But this software will be performed within a 1 day. Now what it will do? It will be performed similarly. It first not dock all compounds. What it will do? It classify the all compound in two parts. One we call the training part and second is called the test part. It divide the compounds in a two part, training part and the test part. In training part, it generate a QSR model. It generate a based on the machine learning. Now QSR is different, machine learning is different. QSR is an old one. Now machine learning is overcome the QSR. It is an advancement of we call QSR. QSR, in terms of QSR, you can work on 100 molecules, but the machine learning can work on the millions of molecules. So in the training compounds is basically create a models on these particular training compounds. Now once the training compounds, the machine learning models are created, now only the training compound will be docked, not complete. First divide training and test, create a machine learning model on the training. Test compound keep aside. Now training compounds are the docked. Now docking stores and the machine learning stores are correlated. Once they correlated, the remaining test compounds are predicted by these models, and that's why this particular software will dock all the molecules in a single day or within 2 days. But you should have to need a graphics processing unit. You should not have to need we call CPU, rather than you need a GPU, okay, which is more faster than the CPU. So it's not exactly called a docking software. It's a software where you have to perform the QSR first, and then you have to perform the docking with respect to training set. The correlation will be there, and ultimately the prediction of a test compound, their binding score, their binding affinity will be predicted. Now lastly, these are the steps we already done. So similarly, there are a number of more molecules, more softwares are available. These can be open source virtual screening softwares. RTDoc software is available. The CanCDoc is there, and even Vina GPU is available. So if anybody is going to work on the GPU system with respect to Autodoc Vina, this Vina GPU is available, which is more time faster with respect to the Autodoc Vina. Now similarly, the AI and ML in a polypharmacology. So I already told you what is the polypharmacology. It's basically called as a one drug and not a one target, it's a one drug and a multiple targets. So you already shown me or said me about the one example, but there are more examples are there, and one of the examples is the metformin. Nowadays, the metformin is basically anti-diabetic drug, but it's right now in a clinical trial, the phase 3 for the early stage breast cancer. So it's one of the, we call the metformin is different. So one drug is there, irrespective of that toxicity, can be used for the multiple targets. So based on that, we have one server that is called as a polypharmacological server, where you just have to provide a structure. Just provide a structure, it gives you the number of target where that structure will go. Now this is a number of target where the structure can be predicted. You do not have to perform any AI model. Already the number of scientists, number of the chemists, number of we call as a chemoinformatics persons have reported number of databases. Just go on the GitHub and search it. You're getting there. So one of the server is polypharmacology server. You have to provide only structure, and you're getting a probable target where this structure will go. Okay, so that can be based on the reported structure. So you can go with the metformin, you go with any other structure. Even you can go search for the aspirin, you can search for the paracetamol and all. Similarly, we have AI and a drug screening. What the drug screening will be suggest is basically we can predict the additive properties. So one of them server is called as a PKCSM server, where it's going to predict the number of properties, and these properties can be with respect to absorption, it can be with respect to the distribution. So this, if you're going to provide a structure, it's going to predict about the, we call it different with respect to absorption property, whether your drug is going to pass the, we call membrane or not, whether it is a 100% absorption, whether it is a 50% absorption, whether it's going to cross the Kk2 permeability, whether it have a water solubility or not, whether it's a probable or good distribution, whether it's going to bound on a protein or not, that you can handle, whether it is metabolized by the cytochrome P450 or whether it is an inhibitor of cytochrome P450, whether it has a good excretion, what is exactly is excretion, whether it will have a toxicities or not, and even it's going to predict the basically the oral rat active toxicity. So because of the database, because how it is developed, because somebody have given the toxicity data with respect to LD50 from the rat, and that's why it's also going to predict the rate related we call toxicity for your structure. So similarly, the one more is there in case of a deep PK, which is similar to your PKCM, is exactly giving lot of different types of we call that particular these things. And the last part is basically that with respect to the we call the retrosynthesis. Now, if you have a structure, and you want to predict its reactant from you are going to get the reactants are there, and how I am going to synthesize it. So normally a chemistry people what they are doing, they will break the molecule in near to the heteroatoms rather than thinking about what the molecules or the reactants available in a market. So what the software will give us, one of the software is called as a AI Zen Finder software. Now what the AI Zen Finder software will do, it work on again the Monte Carlo tree search algorithm and break down your molecule into the purchasable precursors. So you are getting a molecule, if you are giving this molecule to that particular structure, this software, it gives the number of states, and these states which are given is basically purchasable. So you are getting a reactant which is purchasable, so you directly purchase it, you can synthesize a molecule. Rather than predicting manually by breaking the molecule with respect to be called as a heteroatom, rather than give to this particular softwares, it gives you the reactant based on what exact you are doing with respect to purchasable. So the more detail about this software even available on the YouTube, and the lecture is basically from the Dr. Samuel from the AstraZeneca. The software is basically developed by the AstraZeneca scientists, and it is freely available on GitHub, where you are going to about with the retrosynthesis with respect to the any particular structure. So these are the different tools. So with respect to tools are the drug design, some are the tools with respect to the polypharmacology, some are the tools with respect to the drug screening, and some are the tools with respect to the retrosynthesis. So AI is working everywhere. It's not only working in a particularly for the because the text formats are not working on the computer science. It's basically very useful in terms of a drug discovery. We can ease our task rather than doing the manually. We can be using by this software. If going on the GitHub, you will get almost more than 1,000 and the lakhs of softwares are there. The tools are there, which you can use it for the easiness of your work in terms of your dissertation, in terms of your PhD thesis and all. So the number more these are theirs. Okay. So thank you.
Thank you, Dr. Lohani sir, for sharing your expertise with us today. Now the floor is again open for question and answer session. So if you have any question, please just raise your hand. We will bring the mic to anyone from the audience. Do you have any questions? Just raise your hands. Other participants, it's a humble request to please maintain the silence.
Network pharmacology.
A lot of difference are there. Actually, in case of a molecular docking, you are taking a small molecules, you are going to dock on a protein. But in a network pharmacology, you are going to identify the different targets with respect to your gene, with respect to your protein. In terms of a protein, they are connected with each other inside our body. In terms of signal transduction, not a single protein is working. The multiple proteins are working with each other. If you identify one protein, with respect to what the connectivities are there, what the next to that protein, what the next to that protein, you can identify with a neural network pathway. But in a molecular docking, you identify small molecule with respect to the target. Okay?
Which is best for our chemistry student?
Both are best. If you want to work on a new target, first go with the network pharmacology. You identify a new target, so that if your new target is you, then you can select a library of the molecules from databases and dock on that new target. So you're getting a new molecule for the new target. Okay?
Sir, in the Swiss ADMT tool, their drug likeness properties include the Lipinski rule and other properties like Weber and Bosch. So what is meant by Weber and Bosch drug likeness properties?
Actually, these are the people that developed by the different scientists, and that's why they've given a name. Now consider, if I developed a model, so I give you my name. What you have to do, rather than rely on one software, go with all softwares and try to be average of the school rather than taking a one school. Like you are working on a molecular docking itself. You don't go with the rely on one software. Perform the molecular docking of the same molecule with the two softwares and average the score. Similarly, whenever you go with any property predictions with any server, if the two servers are giving the same property, make average. Because these are the prediction based on the input data, and we don't know what the input is actually, what number of inputs we have given, what numbers of inputs given by the server. Because some server have inputs based on the 500 molecules, 600 molecules, some server have based on the 1000 molecules. Just to make an average, so that you get more data.
Okay, sir, thank you.
Any other question?
हॅलो सर मॉलिक्युलर डॉकिंग केल्यानंतर जो रिझल्ट येतो तर आपण का कन्सिडर करतो त्याचे जे -7च्या वरतीच व्हॅल्यू कन्सिडर करतो की जास्त बाइंडिंग ॲफिनिटी झाली ड्रग्ससोबत का ती कन्सिडर करायची -7च्या वरती?
Because the energy is always in the form of a negative. The first thing, the interaction is basically exothermic, not endothermic. Whenever the reactions are endothermic, that means energies are getting, right? And releasing energy is more better. Now consider the simplest example, which I am always giving to my class. If we stand, how much time we can stand? Max 1 hour, 2 hours. But we can sit around 2 hours, 3 hours. But we can sleep around 8 hours. So that's because energy level is very less during the sleeping time. Similarly, the molecules level, they interact to each other whenever they are releasing more energy. So more energy is releasing, with more memory. So negative जितना जाएगा उतना ज़्यादा better. Okay.
Thank you, sir.
Any other question? Other participant, please let her speak.
Good afternoon, sir. So my question is, what is the role of machine learning in de novo technique, de novo drug design?
Again, based on the expansion, if you have a simplest pharmacophore, now consider you have only benzene ring. If you see this benzene ring in a particular form of a protein, और protein में हमने उसको dock कर दिया, एक particular जगह पे dock हो गया है, consider it's dock on, but the protein have a biggest binding sites, right? So you can expand the benzene structure with respect to another connectivities. So you can put up the one side OH, another side CS3, expand with the chain. So this is because the de novo. So in terms of AI, it can predict what the different connectivity with your pharmacophore is there with respect to the protein binding site. Because now consider every lock have a specific key, right? So similarly, every protein have a specific we call structure. and based on that structure, prediction can be easily done by the AI rather than the molecular domain.
And sir, can AI predict the success rate of clinical trial?
Yeah, definitely, it can predict. Because nowadays, the expansion is going on that we call the pharmacology or pharmacology matrix is there, which is basically working in US and European countries, not in India. They have a models, they have a software which is predicting the clinical rate trial success. But we are doing only on the preclinical. If any software is there, if you providing the data of the clinical trial to the software, it can predict a model, it can develop a model, and the model can predict a new clinical trial success for another molecule. That will be possible.